Here we go. So by way of introduction, I'm going to talk a little bit before we get into the main topic, which are sort of digital twins of the eyes and brain. I'll give you a quick kind of high-level overview of the kind of stuff we do in my lab because it's pretty diverse. And because I'm new here, these are things you should come talk to me about sometime if you're interested in them. So we spend a lot of time trying to understand the learning rules of the brain, in particular to understand how brains solve the credit assignment problem, which is to say, how does every individual synapse in the brain know if it should get stronger or weaker when the animal as a whole makes some kind of error? We spend a lot of time making computational models, doing experiments in collaboration with the Allen Institute for Brain Science to try to understand that problem. And that's a really long-standing thread in the lab that is not part of today's talk. We also spend a lot of time trying to take ideas from neuroscience to make new and better machine learning algorithms to solve different kinds of problems related to sort of perception and decision-making in artificial systems. And finally, the thread I'll talk about today is a sort of computational neuroscience applied specifically to problems in visual neuroscience and ophthalmology, which gets us to this theme of digital twins of the eyes and the brain. And I'll mention if you've heard me speak on this topic before, some of this will be stuff you've heard before, so please bear with me. I'll give you some updates on what's new, and then also please prompt me on it, because we do have some, I think, fairly exciting new developments along this theme of research, although we've been at this particular topic for a while. Yeah. All right, so what do I mean by digital twins, and why are these good things to build? The first thing to note is that a digital twin, in my sort of terminology, people use this phrasing a lot these days, but it's really just a simulation. We want a computational model that will simulate, let's say, the eye, take in movies, predict all of the retinal ganglion cell responses that the eye generates to that movie. And there's lots of things we can do with these. I don't actually have a lot of slides on applications, but I'll mention one thing that is in a grant proposal I submitted last week to the Bright Focus Foundation, is that we can use these digital twins to develop new diagnostic tools for things like glaucoma. The key observation there is that in glaucoma, retinal ganglion cells die, but they don't all do so at the same rate. In particular, the larger bodied parasol cells succumb earlier than the smaller bodied midget cells. Other work shows that off-type ganglion cells die before the on-type ganglion cells. And so if you can design a vision test that is specific to a certain cell type, then you can have hopes of doing earlier stage diagnosis. The nice thing about these digital twins, they take in movies and predict what all the different ganglion cell types do. That gives you an opportunity to synthesize pairs of movies that can only be distinguished based on the activity of one cell type. Take a movie, modify it so that all of the cell types except one respond exactly the same way. So it's a metamer for those cell types. And then the one cell type you want to target, you try to maximally modify that cell type's activity in response to the second movie. Gives you a pair of movies you can only tell apart with one cell type, give you a hypersensitive diagnostic test. So this is, among the applications. There are probably many, many more you can think of that grow out of having really good simulations that can predict the way the eyes respond to natural stimuli. Okay, so then we get to the question of how, and there really, I think, two competing approaches to trying to make simulations of biological systems. The first, which has become really, really popular in the past few decades, is to just sort of I say it's a bit pejoratively, but kind of throw lots of machine learning at the problem and hope it works. In other words, use really abstract computational models that could learn to do basically anything, and then have the thing that they learn to do be to take in movies and predict ganglion cell responses. The other scheme that was much more popular back when I was a grad student and exemplified by the Blue Brain project in Europe was this biologically detailed approach where the idea is that you might measure everything you can think of and measure everything that you can about the biological system, put it all into a big simulation and hit go and hope that it works and does the same things that the biological system does. And the the point that I'll first, and I'll make this argument brief because I've talked about this a few times before, but the sort of a big theme in my lab lately has been to try to make a hybrid approach that combines these two things, where we can basically put in all of the biology we know, and then use machine learning to fill in the gaps that we don't yet know, and end up with simulations that work well, but are constrained by as many biological measurements as possible. To sort of highlight that, lots of people have worked on this biologically detailed approach, just show you a couple of papers, and then I'll emphasize their key failure point. This is sort of a nice piece of work from the Allen Institute for Brain Science that I'll talk about more in a minute, applying this idea to visual cortex. And the other paper shown here is really the main scientific product that came out of the €1 billion spent on the European Blue Brain project, or the larger envelope called Human Brain Project, which is a simulation of a little chunk of mouse somatosensory cortex that didn't quite work. And maybe I'll just leave it at that. So what's the key failure mode? Well, here's an example. This is a picture from the paper from the Allen Institute trying to make a simulation of a column of mouse primary visual cortex. These are some pictures of all of the beautifully anatomically reconstructed neurons that they trace and did electrophysiology on those types. And I can make a really hyper detailed simulation of what all of these cells do in V1. And then, of course, if you want to simulate the way the visual system responds to stimuli when your eyes look at something, then you need to know what the inputs are into V1 that come from the visual part of the thalamus. And in this case, they put a linear filter on the movie that is supposed to describe everything that happens in the retina and in the LGN that gets these inputs into V1. So you have this really, you could have a perfect model of V1 here. And you would still probably get very bad predictions for how the visual part of the brain responds when the animal looks at movies. And that's because the inputs are wrong in detail. Right? You've got an overly simplified input tacked on to this thing. And so the key kind of failure mode for the biologically detailed approach is that if there's something that you didn't measure or that you couldn't measure, this system's likely to fail because you're relying on getting all of those details right in order for the simulation to work. And there will inevitably be something you either didn't measure as well as you thought, or couldn't measure. And so the scheme fails. The flip side is to just try to use machine learning and to note that big artificial neural networks are what are called universal function approximators. It means that feed-forward artificial neural networks, the kinds of models we make in my lab and lots of others, if it's sufficiently large, it can learn to copy any input-output mapping you can think of. Those are what mathematicians would call a function. So some rule that takes in some input and generates some output. Any such rule can be approximated arbitrarily well with artificial neural networks if you make them sufficiently large. The main challenge, of course, is training them. So how do you learn the right function from some data? And there's lots of work spent trying to address that problem. But nonetheless, one approach to digital twins, super popular these days, is to give up on the biological details and just use really abstract machine learning models. And just make a big neural net, take in some movie, predict RGC responses at the output. Lots of labs, mine included, have spent a lot of time doing this. This is a paper from my lab applying this idea to primary visual cortex of monkey. Paper next to it, some older work from Steve Backus and Surya Ganguly up at Stanford applying this to salamander retina. The short answer is that it kind of works. You can train neural nets to take in movies, predict neural responses. But this approach fails for a few reasons. The most significant to me as a scientist is that it lacks biologically identifiable parts. And want to know, in addition to being able to solve, let's say, applied problems like making diagnostic tools, I also want to know how the thing works and why it's put together the way it is and why those details within it contribute to function. You lose that in the abstract ML approach. The other thing that sort of fails in the pure ML approach is that the models can fail to generalize to new conditions. So if you've trained your model always on data where the eye was presented with relatively dim stimuli, so low light levels like you might encounter in the nighttime, and then you try to predict the way the neurons in the eye will respond under bright lighting conditions like those seen during daytime, the model's likely to fail because it hasn't been exposed to those kinds of data in order to learn from. Yeah. And the hope is that we might be able to solve those problems by making a kind of hybrid model that lies between these two extremes, which is something I spent a lot of time developing in my lab, where we kind of get the best of both worlds by, again, putting in all the biology we know of, and then using machine learning to kind of fill in the gaps and get models that work well, that have parts that we can identify and study, and then that contain some of the same, let's say, adaptational mechanisms that allow the model to adapt to changing conditions the same way the real biological system does. Okay, and this is just some propaganda to suggest that maybe you get everything when you do this approach. So how do we do this? And I'll show you some data and describe actually some work that a student in my lab came by all excited to tell me about this morning. So this is like really active area of investigation. But the basic idea is that machine learning models like the ones that are used so much in technology applications are really modular. That's one of the things that makes them easy to work with. So when you go to program a big artificial neural network, let's say for something like object recognition in images or text prediction to make something like a chatbot, you don't actually, as a programmer, interact with all of the sort of gory details that are within all of the artificial neurons and the connections between them. Instead, a lot of that has been abstracted away, and most programmers interact instead with what are called layers. So you might take a convolutional layer and layer norm, and then stick it onto an all-to-all layer, and then stick on an LSTM behind it, and end up with something that is sort of big and complicated, but that's made out of parts that you stick together, kind of like Lego pieces in a Lego set. And what's really led to a lot of the big advances in machine learning has been to make nice sort of backends that you don't see as a programmer, that make all of those pieces fit together so that the model can be trained to take in some data and perform some kind of task. So the sort of behind the scenes stuff of doing these like symbolic differentiation calculations to allow the models to be trained, you don't see. All that you really see is, for most programmers, is the sort of module, stick one thing behind another, and then run it and hope that it learns to do your task. And with that analogy in mind, what we've started doing basically of making like new custom Lego pieces to put in the Lego kit. I'll show you some work describing a photoreceptor layer. So literally a little computational model of the signal transduction cascade in photoreceptors that we can stick on to the front end of these models. We've also, this is the work that my student Fazal kind of solved a bunch of in the last week and described to me this morning. Got a nice back end to these models that implements the Hodgkin-Huxley equations in a trainable manner that we can propagate our gradients through them for the aficionados in the audience. And what does that mean? Basically, means we can train the thing end to end, so we can learn the details of ion channel densities and the HH models and all the other parameters all at the same time. So the model can take in movies and predict ganglion cell responses. Okay. So that part of the motivation for the photoreceptor model and the argument for why it's useful comes back to this question of generalization. I think I told you we can train models pretty well to take in movies and predict neural responses in the visual system. I also told you that the machine learning approach fails because the models don't always generalize well to new conditions. And to make that argument more concrete, going to show you what happens if we take the same kinds of models that were in these previous studies that seem to be pretty successful at predicting visual responses. And then we just change by a little bit the experimental conditions. And the motivation for this is that in most visual neuroscience experiments, especially the retina experiments done in the Eye Institute here, the light levels are very carefully controlled. So we don't allow there to be, say, a door opening and closing as someone walks into the lab that lets a huge bright amount of light come in. Instead, trying to very carefully control and avoid for that. Out in the real world, for contrast, brightness continuously changes and by large amounts. This is a recording done by taking this device called an optical power meter. The technician, Maria, just walked around the Duke campus and recorded basically brightness on the vertical axis here as a function of time while she walked around. And you can see there are these really frequent and large changes in brightness level, like a couple of log units, 100. Effectively, as she walks from a shadowy section, like under the shadow of a building or shadow of a tree, you walk out from shadow, it's 100 times brighter instantly. And that happens constantly as you walk around. Or if a cloud comes in, blows over your location, all of a sudden it's like 100x dimmer than it was a moment ago. Of course, you don't notice this for the most part because your visual system's really good at adapting very rapidly to these changing lighting conditions, so that even over the 12 log units of variation in light level that you experience every day, you have relatively good visual perception. Okay, so to understand if this is like an issue for the kinds of models we've been building, we took the same movie, which in this case is sort of little flickering white and black checkers. And we presented it to the retina at 3 different light levels shown here. These units of R star are a measure of like the rate at which the photoreceptors capture photons. And these light levels kind of span sort of nighttime, fairly dim conditions, 30 R star, 3 R star, and then 0.3. In all cases, the data are collected with shown on the far right of the screen, where the retina, in this case a monkey retina, is laid onto a bed of electrodes under basically a video display with some optics to project the video onto the retina. And then a movie is put on the video display while the spiking activity of the ganglion cells are recorded by the multi-electrode array. So you then get a data set that tells you what all of the measurable ganglion cells do in response to the movie. And again, this experiment was repeated in Greg Field's lab, was repeated at 3 different light levels with the same movie stimulus. We then took the model, the sort of classic type of models from these previous studies, so sort of the normal off-the-shelf machine learning models. And we trained the model to take in the movie from 2 of these light levels and predict what the ganglion cells should do. And then we tried to test it on the 3rd light level to see if it could generalize and figure out what's supposed to happen under slightly different conditions. And the short answer is no. It just doesn't particularly work. So here are a bunch of plots that will kind of walk you through. The top is a description of the model architecture, sort of a fairly straightforward feed-forward neural net, takes in a movie, spits out predictions of the ganglion cell activities. This is an example for one cell where the recorded neural activity is in red. The model prediction is the colored curve, and this is for a segment of the movie that was not used in training the model. So the model couldn't have just memorized this mapping between the stimulus and the neural response, but nonetheless can do a fairly good job in predicting this neuron's activity in response to the stimulus. Fraction Stein variance in the 90% range. The same is true at the other light level on which the model was trained. And then the 3rd light level, shown here on the far right, at which the model was not trained, it does very poorly. And in particular, you probably noticed that part of what's happening is that the model's predictions are too fast. So the maroon curve, which shows the model's predictions of what's supposed to happen, tend to precede increases in the neuron's activity because the retina gets slower when the lighting conditions are dimmer. There are other more profound changes, like the fact that whatever's happening at this time in the movie, the model thinks the neuron should respond to it, but the retina, this ganglion cell really just doesn't. Whereas at the higher light level, that particular visual feature did drive the neuron. So just a second, Dean. So there are changes in both the latency and changes to a certain extent in the stimulus selectivity as you change the light level. Yeah, go ahead, Dean.
If you train on 30.3, does it generalize to 3?
Yeah, so all the different permutations of training and testing light levels are included in our paper. The best of those conditions is the ones where you train on 30 and 0.3 and test on 3 because you're interpolating between sort of known training data points. And the worst case is this one where you try to extrapolate to a lower light level. Yeah, good question. Okay. And then these data over here kind of summarize across all the ganglion cells in this data set at the training light levels. You get, in the 80% range for fraction explainable variance for doing predictions on new movies, not the segments used for training the model. And then at the new light level that the model doesn't know about, you do like much, much worse, 24% FEV. Yeah.
No, this is just a detail, and I can't quite see with enough resolution, but are the model predictions across those 3 conditions actually identical?
Yeah, they are. No, this is, yeah, and this is exactly the idea, which I was gonna talk about in more detail in a moment. So what's happening? Like, why is it that the predictions are okay at these light levels and really bad at this one? Basically, at the 30 and 3 on the start light level, the difference between them is not that significant. So the retina adapts a little bit, but not very dramatically. Between 3 and 0.3, the adaptational change is much bigger. So the differences, the differences in these wave curves tell you how much the retina has adapted and generated responses as you change the light levels. And the adaptation is pretty significant here, and the retina is slowing down. The model here has no awareness of the fact that the light levels are changing. In particular, that's because right here at the front end, there's this layer norm, ln, that basically normalizes the amplitude of the inputs so that the model, as far as it's concerned, is seeing exactly the same movie in all these cases and outputting exactly the same prediction. If you don't put layer norm at the front, then the whole system just completely breaks. And that's because your model is, let's say, used to getting inputs that are order 1 in pixel value, and then you go down by 100-fold in light level, and now those inputs are really tiny, and you basically just get zeros for the model outputs. So there's no solution that we could come up with within the sort of realm of normal ML models to get something that made any useful predictions across light levels. This was the best we could come up with, but it has this flaw now that the model is unaware of the changing light level. And so it cannot at all capture the retinal adaptation. Yeah, really good question. Yeah.
If you don't do single cell left, but rather looking at the population, do you reach the same conclusion the model trails?
Yeah, so in this case, here the model is simultaneously predicting the activity of all 37 of these ganglion cells. I've chosen one to highlight here. But the model outputs 37 different predictions at once, one for each ganglion cell. And for each of those cells, we can ask how good the predictions are. That's the numbers in this proximate script plot. And so it is done at the population level. Yeah. Okay. And then the sort of solution that Alex's question kind of points us towards is, well, maybe if we make the model sort of aware of how the retina is supposed to adapt as the light level changes, maybe it can do a better job. So thankfully, we're not like completely in the dark about how well, no pun intended, but not completely in the dark about how these parts of the retina are supposed to function. And so we know a fair bit about the different retinal cell types and their biophysics, starting with the photoreceptors, which, as you'd all know, measure light coming in from the environment. They do so with this signal transduction cascade that sort of starts with this cis to trans photoisomerization of opsins, and then it kicks off this whole G protein cascade, leading to ion channels opening and closing in the cell membrane. And this process itself has sort of some complicated dynamics, but it includes in blue this feedback pathway that allows the retinal photoreceptors to adapt to changes in the light level. Effectively, as calcium starts to build up in the photoreceptor. So what we did to sort of give our models some knowledge of this process is we took the signal transduction cascade, described it by a big set of coupled differential equations that allow us to calculate what the photocurrents should be for any input sort of rate of photons coming in and hitting the photoreceptor. We then made that into a custom PyTorch layer that's kind of shown here schematically in red, and stuck that like photoreceptor Lego piece on the front end to our big artificial neural network. And then once we did that, we repeated the experiment where we trained the model on data from 2 light bubbles, and then tested it on data from the 3rd light bubble. And in this case, with the photoreceptor front end, you get performance at that 3rd, like new test light level that is still worse than at the 2 light levels the model's seen during training. So those again, kind of 80% FEV. At the lower light level, the model's never seen before. You get 54%, but that's much better than you get with the model that does not have the photoreceptor front end, where that drop off in performance for the new light level was down to 24% instead of 54. So we can sort of recover something like half of that generalization error. So the error in going to a new light level, we get something like half of that recovered already by just using the photoreceptor front end. And of course, there's lots of other retinal cell types whose detailed biology we have not modeled that kind of fill in the rest of this 30% that we're working on. Okay. And then the other point that I think is important here is that the issue of adaptation isn't just one where you go from sort of one constant light level to another constant light level. Adaptation is sort of a constant ongoing process, as you might have imagined from that trace of light levels over time in the outdoor environment of the Duke campus. And so the next experiment we wanted to do is to see if we could capture some of that, let's say, more dynamical adaptation in our model. And so to do this, we replaced those checkerboard stimuli with this stimulus, which is just a grayscale natural image. You can see it's like grass next to a pond, and the image moves around, simulating the saccade trajectory of an individual looking around the environment. Right? So as you look around the world, even if there's nothing moving around you, the location at which some part of the world is imaged onto your retina will change as the retina shifts around. And this mimics that sequence of fixations. This is data collected from Mike Mnookin's lab up at U Washington. And then the important point to make here is that this has that same property of changing the local light level experienced by each RGC. You imagine your ganglion cells sitting here on the retina. Well, as the eye moves around, as the image moves around on the retina, sometimes a dark thing is going to fall onto your receptive field. And other times, some bright thing is going to fall onto your receptive field. And so you can very rapidly experience these brightness fluctuations that, again, can be, orders of magnitude in size and extremely frequent. And this scatter plot just shows the model of prediction performance for the 2 types of machine learning models I've described to you. Vertical axis is the model that has the photoreceptor front end. The horizontal axis is the conventional convolutional neural net. Each circle represents one ganglion cell from this data set, and the values show the prediction performance. And across the board, we do, I guess not for every cell, but we systematically do much better at predicting neural responses, even to this stimulus at a constant mean light level. We do much better at predicting responses with the photoreceptor front end than without it. And that's because, again, photoreceptor model can capture the local adaptation changes of subsets of the photoreceptors as they're experienced to dark or bright stuff. Okay, so this paper has been published. It's kind of old news, but I think still stuff I'm kind of excited about. If you want, you can download these models from Saad's GitHub repo. Saad Idris is the postdoc in my lab that did this work. So you can stick these onto the photoreceptor model onto your own machine learning models and kind of play with it as you wish. And so the parts that I've talked about that we've kind of done so far is to make like the photoreceptor Lego piece, stick it onto the front end of conventional abstract machine learning components. Where we're going next, and like now actively, is to replace more and more of these like abstract machine learning like with more custom trainable neuron layers. Most recently, we've made like taken yeah, I should say we've made that from scratch. We've taken differential simulation of the Hopf-Huxley equations that we've implemented in the same framework. And then we can stick it onto the back end of our model and train the model to take in movies and predict the actual voltage traces of the ganglion cells. And then, the thing that we struggled with a long time and have just solved much to my delay is to actually be able to propagate gradients all the way through the HH model. In other words, be able to train the ganglion cell detailed biophysics output model along with all the stuff that precedes it so that we get good predictions. For the ML or even the modelers in general in the room, that's kind of tricky because the spiking nonlinearity is like very, very sensitive to inputs, and it makes the issue of calculating what change in something before the spiking should have happened in order to change the voltage output. It makes that process really, really sensitive, and so the details of getting that right were quite tricky.
So when you do that, are you training the HH parameters for each ganglion cell independently, varying across ganglion cells?
So at the moment, the stuff that we have got to work like in the past few days that I heard about this morning is training this to do just one ganglion cell. Where we're going to go next, right, we have all the data and the sort of framework set up to predict many ganglion cells at once. The immediate next step is to have different HH parameters for every ganglion cell, because that gives us the most likelihood to make things fit right. And then the hope is that we'll be able to later have common sets of parameters for each cell type. So the paracel cells with their larger cell bodies will have different parameters, different capacitance of the cell membrane because they're bigger than the midget cells with their smaller cell bodies, and to have cell type specific sets of biophysics parameters, but we haven't done that yet. Good question. Okay. And then, okay, I'll talk about some other stuff in a sec, but I want to just emphasize a higher level point that didn't make it into the slide deck, which is I think the same approach could probably be applied elsewhere in neuroscience. And what I'm hoping to do in the next small number of years is again replace all of this with biophysics models to have a real trainable digital retina that has all the right cell types in it. Where I'm hoping to go beyond that is to make like a digital brain. We know a lot about the biophysics of neurons all over the brain. We know what a pyramidal cell looks like and what it's supposed to do. We know what a VIP interneuron looks like and what it's supposed to do. And so there's in principle no reason we can't extend this framework and make a fully trainable brain simulation that I think could be extremely rad. Right. And then part of how we're starting to scale this up towards that goal is to note that the experiments I've shown you have been done on a single retina. So taking the movie and predicting one ganglion cell's responses. The amount of data you can collect from one retina at a time is limited. The duration of the recording is limited by how long the tissue lasts on the array. But we started collaborating with EJ Chisholmitsky up at Stanford. EJ has been collecting these types of multi-electrode array recordings for decades. He won't like me to tell you how many decades, so I won't, but it's several. And so he has something like 1,000 of these data sets on disk from monkey retina and 10 from human retina, of which about 100 have been carefully, 112 have been sort of carefully spike sorted and pre-processed, so that they're ready to do these kinds of analyses on. So what we want to do is to be able to train the same model, not just on one retina, but on all of them, to get one kind of master digital retina that has learned everything it can from the whole cohort of individuals whose data have been used in training the model. Good, right? And there's like 1,000 more monkey retina, 10 more human retina available. This immediately poses a challenge because we need to be able to have the same model make predictions for any retina. And if your model just takes in a movie and predicts the activity of ganglion cell 1 or ganglion cell 2 or ganglion cell 3, then if I want to try to make predictions for a new individual, now I need to know which ganglion cell in that individual is ganglion cell 1 or 2 or 3. And there's not like some label on the cells that says that this cell from your retina looks like this cell from my retina. The sort of nice attribute of the retina that allows us to solve this problem, though, is that the ganglion cells tile the retina. So each cell type, here on and off parasol cells are shown. These little outlines in these cases are the receptive fields of those neurons. And they tile the retina, so they sort of cover in space the back of the retina. And it means that we can adopt a new approach, which we started doing back in February, where instead of taking in a movie and predicting the activity of each ganglion cell, we take in a movie and we predict a spatial map, where the value of that map at some location says this should be the activity of a cell at this location. And then what we do is we have one such map for every ganglion cell type. So our model takes in a movie, predicts the activity of each ganglion cell type at each location. This is just a picture of a spatial map. And once we do that, we can start to make zero-shot predictions. This is an example of a model that's been trained on data from some monkeys, now predicting the activity of a ganglion cell from a new monkey that's shown here in blue. The orange is a prediction that comes from the model trained on other monkeys. And there's a few things to note. One is that the prediction is not perfect. The blue doesn't match the orange. The second thing, which encouraged me when we started to see results like this, is that they were at least relatively correlated. So the model predictions tend to go up and then real ganglion cell activation goes up. And so it's not quite as hopeless to do this cross-individual generalization as it might have initially seemed. And in fact, it more study and a little bit better models down the line show us that we actually can do almost as well under the right with the right set of tricks, almost as well at the zero-shot generalization as we can if we make a specific model for each individual. I'll walk you through this plot because there's a lot of stuff happening. So here we started by training a model on like 75 monkey retinas, and we're testing on 23 others from new individuals. And the dashed black line on this plot is the prediction performance of that zero-shot prediction. So if you don't know anything about the monkey for whom you're making the predictions, how well can you predict ganglion cell activities? The let's say red curve here is the prediction performance if you allow the model to be fine-tuned, so you allow it to be updated with data from the monkey for whom you're making the predictions. You start with the model that's learned from every monkey, update it a little bit with data from the new monkey, then test how good it is on that new monkey. That's the red curve as a function of how much data was used from that new monkey for the fine-tuning. So right, the zero-shot predictions are totally flatlined because they don't use any data from the new monkey. The red curve asks if you fine-tune the model. And so the difference between red and black is kind of how much you win by accounting for individual variability between monkeys. And it's some, but not a huge amount. And then the other two things I was talking is vector-based model. That's the blue one. That's not my phone, right? No, okay. So the vector-based model is sort of like the one I alluded to earlier, where we train the model from scratch, not to predict these maps, but instead to predict a vector that says what the activity should be of every cell. And that one's obviously trained on just the one monkey. So it starts out being very bad with only a little training data. It's better as you add more training data. And ultimately that's better than these map-based models. And that's because that vector-based model can account for cell-to-cell variability. It knows that one of these ganglion cells might be a little more excitable than another, might by chance have a little bit bigger receptive field than the others, and so on. And so the blue to black gap is, that whole difference is kind of everything that you lose if you ignore cell-to-cell variability and individual-to-individual variability. And it's not zero, but it's not overwhelmingly huge, which got me kind of excited about the prospect for doing some of these zero-shot modeling. Okay, so there's lots of work to do here on the digital retina front. We're like making more ganglion cell types. We're expanding that data set. Mentioned there's like 1,000 of these data sets, but recordings, but they're not all carefully spike sorted. That's work that's ongoing with Vijay Chichalmiski's lab. We're working on improving the sort of zero-shot prediction models. I can tell you about some of that. And then working on using this overarching framework to make like diagnostic tools or camera to retina translators for prosthetics, and all sorts of other fun applications. The final thing I want to talk briefly about, which I alluded to earlier, is this sort of like diagnostic tool, this idea of making cell type specific stimuli. And this is stuff that initially kicked off by Sada Dries in my lab. Now sort of as the torch has been picked up by Nilu Gazavi and Greg Field's lab, and Guillaume Raval in Frederiki's lab up at UWashington. And what we've started doing is taking our predictions and then modifying stimuli to target individual cell types. And part of the reason to do this is that your retina, something you probably don't confront every day, but your retina has about 25 different retinal ganglion cell types, 25 to 30 in the front of the retina, something like 40 in the mouse retina. And each of them responds to different attributes of the visual scene. And so when you think about the optic nerve, those million axons all grouped together, they're not all conveying the same types of visual information to the brain. They're really conveying very different types. So this is just a schematic, or a picture of kind of the zoo of cell types from E.J. Chichalmiski at Stanford, showing receptive fields of a bunch of the different cell types. The numerically most dominant are on and off midget and parasol cells. Those are something like 70% of the ganglion cells in your retina. And then all of these other, let's say, more exotic cell types make up the other 30% or so. And what we've started doing is trying to tweak, to stimuli, and again, sort of customize them to really isolate the function of one cell type at a time. And to show you schematically how this works, the idea is we start by taking a movie and we can pass it to our model and get predictions for all the RGC activities. I'll show you this schematically where we're doing this in the context of only 8 ganglion cells. The idea scales up, but this demonstration is for 8 ganglion cells. The receptive fields are shown here. And there are 4 on-parasol cells that sit shown in green, those are receptive fields. And then 4 off-parasol cells, their receptive fields are shown in red. The game is to adjust the pixel values in this movie so that we can change the activities of the off-parasol cells, but not the on-parasol cells. And that's somewhat challenging because the receptive fields overlap. So we just grab a pixel here and make it brighter, make the on-parasol more active, but the off-parasol less active. So finding ways to modify this movie that doesn't affect both cell types is in principle a little bit tricky. The solution, though, is the fact that we can calculate from the model, we can calculate the sensitivity of every neuron to changes in every pixel value. Mathematically, that's a derivative of the predicted firing rate with respect to the pixel value. And if I can assemble that for all the pixels, it gives me a big long vector in pixel space that says if I change the movie along that sensitivity direction, that's when I would get the biggest change of the neuron's activity. And if I pick a change in pixel values that's perpendicular or orthogonal to that direction, then I can change the pixel values without altering the neuron's activity. And so what we do is we take these gradients, so vector of sensitivity of a neuron to all the pixel values, and we change the stimulus along the sensitive direction for one neuron. In this case, it's neuron A. And we then remove the component that is not along the sensitive direction for the other neurons that we want to leave alone. So we end up finding a direction in pixel space that's completely perpendicular to the sensitive directions for the subtypes we want to leave alone, and has some component along the sensitive directions for the cells we want to change. And that lets us make these changes again in a way that leaves some cells alone and affects others. This is I guess in this case, this is one frame from the movie of the actual change in the stem. And then, we end up with a modified stimulus in the end that can be very subtly different from the initial one, but has been changed in a way that just affects the one cell type and not the other. I think I've got a movie. So here's a movie showing that. The original movie is on the left. The modified one is on the right. The cell receptive fields are kind of down here-ish, and so you'll notice some changes in that location. And sometimes in the movie, although they're fairly subtle, kind of here-ish. After that modification process, we end up with a stimulus that doesn't drive the off peristalsis at all, as far as I'm looking at. So here's like firing versus time for the 4 off peristalsis or on peristalsis demonstration. The neuron's response to the movie is in gray. The model's prediction is the colored curve. And then the model's prediction for the response to the modified movie is in blue. So as far as our model can tell, this stimulus won't drive the peristalsis at all. Or the off peristalsis. When we look at the on peristalsis, the predictions, predictions in green for the original movie and predictions in blue for the modified movie are identical by construction. So we, again, as far as our model can tell, have stimuli that should be able to isolate one cell type and leave the others unaffected. There's tons of work to do in this space. Among other things, we need to validate that these model predictions are accurate for the modified stimuli. So go back to the MEA recordings and show that when we show the one movie or the other, we really do only change the activity of one cell type and not all the others. We also need CNN models that can predict all like 20 to 30 primate RGC types and not just the 4 dominant ones, which is what we currently have. Yeah, so there's lots more work to do in sort of fleshing out this space, but I'm excited about the idea that we can make vision tests that could really isolate, one cell type or another at a time. So I'll wrap up here and mention that we can, use this kind of hybrid approach to make models of neural circuits in the retina. It is possible to do zero-shot generalization, and the fact that the models are mathematically differentiable means we can use them to do this designer stimulus game. And there's lots of people who contributed to this work. And I'll just show you their lovely faces and take some questions.